One of the most common ways to use the knowledge portals is to check whether there is human genetic evidence that supports the potential role of a gene of interest in a trait or disease of interest. Here we'll illustrate the genetic support workflow that can help you do that. We'll use the Type 2 Diabetes Knowledge Portal for this example, but this workflow is applicable to most other knowledge portals. This video will give you a high-level overview, and you can find all the details under the Help menu. In this example, we'll check whether there's evidence for the role of the SYN3A gene in type 2 diabetes. Starting from the home page, start typing a gene name and select the correct one. This takes you to the region page, showing genetic associations and genomic annotations across all phenotypes in the portal, in the region spanning the gene and its flanking sequences. Check the phenome-wide association, or PheWAS plot, near the top of the page, showing genome-wide significant associations across this region. If there is human genetic support for the role of a gene in a phenotype, there will usually be at least one genome-wide significant variant association for that phenotype shown on the region page. The dotted line here indicates genome-wide significance, so you'll want to look for points above it. We see that there is a genome-wide significant association for type 2 diabetes. Next, let's look at the gene page. To get there, go to the top of the region page and click the blue box. Let's scroll down the page a little bit to the first PheWAS plot. This plot shows gene-level association scores for SYN3A calculated using the MAGMA algorithm from common variant associations. A generally accepted significance score for MAGMA results is a p-value under 2.5 times 10 to the negative sixth, and this is again indicated here by the dotted line. So we can see that the SYN3 association does meet this criterion. However, we should remember that a MAGMA significant result indicates that the gene is close to a significantly associated variant and doesn't necessarily mean that the gene itself is causal for the phenotype. Just below this PheWAS plot is another that displays gene-level association scores generated from rare variant associations. The generally accepted threshold for exome-wide gene-level significance is a p-value under 6.57 times 10 to the negative 7, again indicated by the dotted line. So we see that in this case, the type 2 diabetes association does not meet that criterion. However, SYN3A does have a significant association for a related phenotype, 2-hour glucose. To see a simple summary that takes into account both common and rare variation, check the Human Genetic Evidence Calculator score at the top of the page. The score for the most significantly associated phenotype in this region is shown by default. So to change the phenotype, begin typing a phenotype into the box and select the one you're interested in. Here we see that despite the lack of strong rare variant evidence for the involvement of SYN3A in T2D, the overall evidence for its involvement is very strong. Finally, the knowledge portals include expert-curated lists of predicted effector genes for many diseases and traits. To see these, navigate to the Predicted Effector Genes link under the Tools menu. Here you can browse the available lists. In each portal, the lists available are those that make predictions for the phenotypes that are included in that portal. We'll select a list generated by Anupa Mahajan and Mark McCarthy. All of the effector lists allow you to search by gene name. SYN3 isn't included in this list, so let's look at a different gene, PAM. The summarized evidence is shown in the default view, and if you click on the Evidence button, the rows expand to show different kinds of supporting evidence that were considered in making these predictions. All of the details of the prediction method are documented on the Research Method tab. If your gene of interest is included in an effector prediction list, you may find other kinds of evidence supporting its role, such as mouse mutant phenotype evidence or EQTL evidence, evidence types that are not yet included in the knowledge portals. We're happy to help you evaluate the evidence that you can find in the knowledge portals. Please contact us.